Every single person listening to this right now has, on average, about 88 genetic mutations actively changing how their bodies build proteins.
Right, yeah. And up until literally today, almost half of those changes were just, well, completely invisible to science.
Which is just a staggering blind spot. I mean, would you really consider that these unseen mutations dictate the fundamental building blocks of human biology?
Exactly. We've basically been trying to read the instruction manual for the human body while, missing half the pages.
Missing half the pages. That is exactly what we are looking at today. Welcome to the Deep Dive. We are unpacking a brand new, highly complex methodology paper that just dropped in Nature Methods.
Yeah, March 30th, 2026.
Right. And it introduces this groundbreaking computational tool called longcallR.
Which is such a massive leap forward.
It really is. It basically acts as this hyper-advanced translation engine for our genetics because, it solves this massive headache in biology, which is how to accurately read these long, messy stretches of RNA.
So we can finally connect a specific genetic typo directly to, well, a physical change in how our bodies function.
Exactly. So our mission today is to break down exactly how this algorithm works, how it performed against our absolute gold standards, and, what happened when researchers unleashed it on the human population.
And found all this unmapped dark matter in our genetics. It's fascinating.
It really is. So, okay, let's unpack this. To really grasp the magnitude of what longcallR does, I feel like we have to start with the tech it was actually built for, right?
Yeah, we have to talk about long-read RNA sequencing. For a really long time, the standard in biology was short-read sequencing.
Right.
But now, technologies from companies like Pacific Biosciences, so PacBio and Oxford Nanopore Technologies, they're just completely changing the landscape.
Okay, let me see if I can visualize the difference here for everyone listening. Short read sequencing always sounded to me like trying to understand a massive 1,000-page novel, but the book has been run through a paper shredder.
Oh, that's a great way to put it.
Like, you have millions of tiny strips with maybe 3 or 4 words on them. You can sort of guess the overall plot by looking for overlapping words, but you lose the whole context of the chapter.
That is a perfect analogy. You get the raw data, but you completely lose the structure. Long-read sequencing, on the other hand, is like finally getting whole, intact paragraphs.
Okay.
You can read a sentence from beginning to end without having to guess how it connects to the next one. And because you have the whole paragraph, you can finally see how multiple genetic variations, what we call SNPs, or single nucleotide polymorphisms.
How they relate to each other.
Exactly, how they relate to each other on a single continuous transcript of RNA.
And that relationship is critical, right? Seeing them together.
Absolutely. It allows for this concept called haplotype phasing.
Phasing.
Yeah, phasing is basically how we determine which specific parental chromosome a transcript actually came from. Like, did this entire sequence of variations come from your mother's side or your father's side?
Which you can't really do if it's all shredded up.
Right. You can't figure that out easily if the sequence is just tiny disconnected pieces.
Okay, wait a minute. Let me just pause here. If finding these genetic typos in RNA is this messy, why not just sequence the DNA?
What?
Like, isn't DNA the original blueprint anyway? Why go through the massive headache of looking at the messy RNA?
That is honestly the core question of why the entire field of transcriptomics even exists.
Okay.
Think of DNA as the static blueprint locked away in a library. It tells us what the instructions are. But RNA is the active construction site.
Oh, I like that.
Yeah, RNA tells us what instructions are actually being used, how loudly they're being shouted to the builders, and how those instructions are being cut and pasted together to build, different versions of a protein.
So if we only look at the DNA blueprint, we just completely miss the actual real-time activity?
We miss the dynamic activity of the cell entirely.
Okay, so the DNA says, build a window, but the RNA tells us if the cell is building one window, 1,000 windows, or if it, decided to splice the instructions and build a skylight instead.
Precisely. And that cutting and pasting process is what we call splicing. But here is the major problem.
Yeah.
Analyzing RNA is incredibly difficult because it's just so messy.
How so?
Well, first, you have massively uneven coverage. Some genes are expressed really loudly, meaning the cell creates thousands of RNA copies for the sequencer to read.
Okay.
But other genes are very quiet, maybe producing only 2 or 3 copies total.
So it's hard to hear them over the loud ones?
Exactly. Second, that splicing process creates alignment errors. When the computer tries to map the spliced RNA back to the original DNA genome, It just gets confused around the edges where the cuts were actually made.
Like it's trying to map a customized edited document back to the original draft, and the software just trips over the edits.
That's exactly it. And the third hurdle is perhaps the most difficult for algorithms to handle, which is post-transcriptional RNA editing.
Okay.
Specifically, this phenomenon known as A to Y editing.
Wait, A to I editing, what does that mean in plain English for us?
Right. So it means the cell intentionally changes the RNA after it copies it from the DNA. The cell alters an adenine base, an A, to make it look and act like an inosine, an I.
Really? Just intentionally?
Yes. It's a deliberate functional edit made by the cell to create more diverse proteins.
But to a sequencing machine, an A suddenly turning into something else, I mean, that looks exactly like a random genetic mutation.
Yes, exactly. The sequencer sees a typo. All the standard algorithms see a typo. But it's not a mutation you inherited from your parents. It's a deliberate edit happening in real time.
Wow.
And distinguishing a true inherited genetic mutation from a deliberate cellular edit is just phenomenally difficult.
Okay, let me just do a quick recap here just to make sure we're taking you, the listener, along with us before we dive into the algorithm itself.
Good idea.
So if I'm tracking this, point one, we need long-read RNA sequencing because it gives us the full paragraphs of what our cells are actually doing. Point two, actually reading those paragraphs is a nightmare because the coverage is uneven, the splicing confuses the map, and the cell makes deliberate edits that look exactly like inherited mutations.
Yes.
And point three, our current tools just aren't smart enough to handle all that noise.
That is the exact landscape of the problem. Current tools either lack the accuracy or they simply cannot perform the complex allele-specific analysis we need, and that is exactly why the researchers had to build a completely new kind of computational machine, longcallR.
All right, let's look under the hood of this machine then. From reading the paper, longcallR isn't just like one block of code. It's structured like a pipeline with 3 distinct brains working together.
Yes, 3 modules.
Let's start with module 1, which they call longcallRNN. This is a neural network, right?
It is. It uses a deep convolutional neural network, specifically an architecture called ResNet-50. And what is truly brilliant about this first module is how it interprets the data.
How so?
It doesn't look at genetics like a string of text. It literally turns genetics into a picture.
Wait, what? How do you turn a sequence of biological letters into an image?
It creates a visual matrix.
Yeah.
So imagine a picture that is 41 pixels wide.
Okay, 41 pixels.
The center pixel is the candidate genetic mutation, the SNP we were looking at. The 20 pixels to the left and the 20 pixels to the right provide the immediate genetic context surrounding that mutation.
So it's looking at the neighborhood.
Exactly.
Yeah.
But it's not a flat black and white image. It's a tensor, which you can think of as a deep, multi-layered image. It actually has 7 different color channels.
So instead of just red, green, and blue, this image has 7 layers of biological data stacked up. What are those layers actually capturing?
They capture every nuance of the sequencing read. So you have a layer for the base being read, a layer for the original reference sequence, a layer for any insertions the sequencer found.
So that's the physical sequence data.
Right. Plus a layer scoring the base quality, one for mapping quality, one showing the read strand orientation, and one showing the transcript strand orientation.
Okay, so the AI is looking at this 41 pixel wide, 7-layer deep stack of data. What is it trained to actually output?
It acts as an image recognizer. It analyzes that rich visual pattern and makes 2 predictions. First, it predicts the specific genotype. For example, is this position a combination of an A and a G?
Got it.
Second, it predicts the zygosity state.
Zygosity. Let's unpack that because the paper mentions 5 different states this neural network can output.
Yeah, so zygosity just describes the similarity of the genes you inherited from your parents. The AI can output 0-0, which means there's no mutation, just a match to the reference.
Okay.
It can output 0-1 for heterozygous, meaning you have 1 mutated copy, 1 normal copy. 11 for homozygous, meaning both copies are mutated. 12 for biallelic, meaning both copies are mutated but in different ways.
Right.
And this is the crucial part. It has a 5th, highly specific category just for that pesky A to I RNA editing we discussed earlier.
Oh, wow. That is clever. So it doesn't just try to blindly filter the RNA edits out. It explicitly learns the unique visual signature of a deliberate cellular edit versus a true inherited mutation.
Exactly. It uses the deep visual context of those 7 layers to say, hey, this isn't an inherited typo. This is the cell editing itself. Flag it and separate it.
That's brilliant.
And once we have these verified true SNPs from the neural network, the data flows into Module 2, longcallR phase.
And this module is written in the Rust programming language, right? And its entire job is basically matchmaking.
Matchmaking in the sense of parental lineages, yes. It needs to take all these true SNPs and mathematically sort them. It wants to know which of these variations came from your mother's chromosome and which came from your father's.
Right.
So it builds a probabilistic mathematical model to sort them into two distinct buckets.
But how does a math equation know where to start? I mean, if you have thousands of variations, it can't just guess randomly.
No, random guessing would take an astronomical amount of computing power. Instead, it uses a technique called heuristic initialization, which means, think of it like building a massive jigsaw puzzle. You don't start with the blank sky pieces, right? You find the corner pieces and the highly distinct edge pieces first.
Oh, okay.
The algorithm scans the data for a few highly reliable, unambiguous SNPs to act as its anchor points.
So it builds a foundation of a few things it is absolutely certain about.
Right. It creates a mathematical graph linking the reads, which they call haplotags, with the SNPs, the haplotypes. Once those anchors are set, it starts an iterative process.
Iterative meaning it repeats.
Yeah. It takes a variant, calculates the probability of it belonging to mom's side, and temporarily assigns it there. Then it checks the overall mathematical conflict in the entire system.
Like checking if forcing a puzzle piece into a spot messes up the alignment of all the pieces around it.
Exactly that. It flips the assignments back and forth, recalculating the probability score after every single flip to minimize conflicts across the entire graph.
Wow.
It keeps alternating thousands of times until the probability converges, meaning the score just stops improving. At that point, the variants are phased. They are cleanly separated into parental lineages.
Okay, let me do another quick summary here. So Brain 1, the AI image recognizer, spots the real typos and throws out the cellular edits.
Right.
Brain 2, the Rust matchmaker, takes those typos and mathematically sorts them into mom's and dad's copy.
Yeah, exactly.
Which leads us to the final step. What does Module 3 do? Because up to this point, we just have a very clean, sorted list of data. What is the actual biological payoff?
Module 3 is the analyst. This is where we run the statistical test to see if these genetic variants are actually doing anything. It looks for two phenomena, allele-specific expression, or ASE, and allele-specific junctions, or ASJ.
Okay, let's translate those. What is allele-specific expression in terms of our body's instructions?
Think of expression as volume. Normally, if you inherit a gene from mom and a gene from dad, you expect them to be expressed equally. A 50-50 split of volume.
Makes sense.
Module 3 uses a statistical model called a beta binomial test to see if that volume is skewed.
A beta binomial test. Let's make that intuitive for you listening. How does that work?
Imagine flipping a coin. You expect it to land heads 50% of the time and tails 50% of the time. If you flip it 100 times and get 90 heads, you know the coin is rigged.
Right.
The beta binomial test does that for genetics. It looks at the RNA copies and says, are we seeing mom's gene 50% of the time, or is mom's gene producing 90% of the RNA?
Yeah.
If it's 90%, that genetic variant is causing allele-specific expression. The variant acts as a volume dial.
That makes perfect sense. And what about the allele-specific junctions, the ASJ?
If ASE is the volume dial, ASJ is the cutting room floor. It tests if a specific genetic typo is physically causing the body to splice the RNA differently, and it uses a statistical method called Fisher's exact test.
Okay, but how does Fisher's exact test work in this context? Let's avoid just dropping the statistical jargon. How does the math actually prove a biological change?
Fair point. It basically sets up a 2x2 grid. On the left side of the grid, you have the parental origin, mom's gene versus dad's gene. On the top of the grid, you have the physical shape of the RNA. Splice pattern A versus splice pattern B.
Okay, I can picture that.
The math simply tallies up the reads. If the grid shows that mom's gene always results in splice pattern A and dad's gene always results in splice pattern B, well, the algorithm proves that the genetic variant is the physical cause of that distinct cut and paste job.
That is incredible. It's literally connecting the abstract genetic code directly to the physical architectural changes in the cell.
It is a beautifully elegant computational pipeline.
It really is. Okay, so bullet summary time again. This 3-part pipeline takes messy RNA. Module 1 turns it into a 7-channel image, so AI spots the real SNPs. Module 2 mathematically sorts variants into lineages. And Module 3 runs these statistical tests to see if the gene's behavior actually changes.
Perfect summary. But, as anyone in biology knows, a tool can look perfect on a whiteboard and completely fail when you apply it to actual messy biological data.
Right, theory is great, but how did they benchmark this? If you build a new ruler, you have to test it against something you already know the exact length of.
Precisely. They benchmarked longcallR against the ultimate gold standard, Datasets from the Genome in a Bottle Consortium, or GIAB.
For those listening who might not be familiar with Genome A Bottle, what makes these datasets the gold standard?
GIAB provides reference genomes that have been sequenced so thoroughly by so many different labs and technologies over the years that we effectively know their exact, true baseline sequence.
Ah, okay.
They are the calibration weights for the scientific community. So the researchers took long-read RNA data from specific GIAB samples and fed it into longcallR to see if it could correctly identify the mutations we already knew were there.
And they specifically tested it on the two major long-read sequencing technologies, right? PacBio and Oxford Nanopore. So let's look at the results. How did it do on the PacBio data?
On the PacBio datasets, longcallR achieved stunning precision. It hit between 98.5% and 99.0% precision in correctly calling these variants.
That is remarkably high. But when I was looking through the charts in the paper, I noticed an interesting nuance.
Oh.
They compared longcallR to a slightly older tool called clair3 RNA. And clair3 RNA actually had a slightly higher sensitivity, meaning the older tool found a higher total number of variants. Why would we want a tool that technically finds fewer things?
What's fascinating here is that understanding the mechanism of the algorithm answers that. LongcallR acts like an incredibly strict bouncer at an exclusive club.
Okay, a bouncer.
Yeah, the older tool, clair3 RNA, is very good at spotting potential variants. But longcallR has that strict second module, the Rust matchmaker. It absolutely requires every single SNP to be perfectly phased into a parental lineage.
So if the math in Module 2 can't cleanly prove whether a variant came from mom or dad without conflict, longcallR just throws it out completely.
Exactly. It sacrifices a tiny bit of total recall, meaning it might toss out a few real variants that were just too messy to phase in order to absolutely guarantee that every single thing it lets through the door is perfectly precise. In diagnostics, a false positive say that telling someone they have a mutation they don't actually have is often much worse than missing a weak signal.
So it's optimizing for absolute trust. If longcallR says it's there, you can bet your house on it.
Precisely.
Now, what about the Oxford Nanopore data? Because nanopore technology works differently and generally has a higher baseline error rate than PacBio, right? How did this strict bouncer handle that noisier data?
You're right, nanopore data is noisier. Because of those higher underlying sequencing error rates, the tool's precision dropped to between 85.8% and 98.0%, depending on the specific data set.
That's a bit of a drop.
It is, however, it performed phenomenally well on Nanopore's newest direct RNA datasets. And the most important takeaway is that even on this noisier nanopore data, longcallR still significantly outperformed the older tools in precision for exactly the same strict filtering reasons.
Okay, quick recap of the benchmarking. longcallR is tested against rigorous human reference genomes. It achieves near-perfect precision on PacBio, and it outperforms competitors because it strictly requires phased data to validate a finding.
Spot on.
Which means the algorithm is highly resilient regardless of the machine feeding it data. So the tool works. It's hyper-accurate. Now we get to the most exciting part of this entire paper. What happens when you take this pristine new telescope and point it at the broader human population?
This is the real biological payoff. The researchers didn't stop at reference genomes. They unleashed longcallR on 202 different human samples sequenced by the Human Pangenome Reference Consortium, the HPRC.
That is a massive data set, 202 distinct individuals from across the globe.
Yeah, they ran the PacBio data for all 202 individuals, specifically looking for those allele-specific junctions, the ASJs.
The cutting room floor changes.
Right. Again, this is the holy grail. Finding where a specific genetic variant is directly physically causing the RNA to be spliced differently, changing the structure of the resulting protein.
And the raw numbers they pulled out of this population were wild. They identified 1,852 genes with highly significant allele-specific junctions.
It's a huge number.
And when you average that out, it means every single person walking around has roughly 88 of these highly specific, genetically driven splicing events happening in their bodies at any given time.
It just shows how incredibly dynamic human biology really is. But the statistic that completely reframes our understanding of transcriptomics is this. Of those ASJ events they found across the 202 people, 46.5% involve completely unannotated novel junctions.
I really want you listening right now to think about what that means. Nearly half, almost 50% of the alternative splicing events this hyper-accurate tool discovered are completely missing from our standard genetic maps.
They aren't in our reference databases at all. GenCode V39, nothing.
This is transcriptomic dark matter.
It is a massive biological blind spot. We've been trying to map the complexity of human disease and cellular function without knowing that half of the specific splicing variations even existed.
But, okay, I have to play devil's advocate here.
Sure.
If you build a brand new software tool, and the very first time you run it on a population, it spits out data saying that 50% of the results are things no one has ever seen before, doesn't that raise a massive red flag?
It absolutely should.
Like, how do we know the software isn't just hallucinating? How do we know these aren't highly confident artifacts or errors from the AI?
That is the exact critical question the scientific peer reviewers would ask, and the researchers anticipated it. They proved these novel junctions are real biological features, not software glitches, by looking at human ancestry.
Oh, how so?
They ran a statistical method called principal component analysis, or PCA, on these specific ASJ genes across the 202 people.
What does a principal component analysis actually do in this context?
It looks at the massive web of splicing data and asks the computer to find the natural groupings. Without giving the computer any geographic or demographic data about the 202 individuals, it asks, do these novel splicing events cluster together in any predictable way?
And what did the natural clustering show?
The data naturally and perfectly separated by geographic populations.
Wait, really?
Yeah. The first major grouping separated the African samples from the non-African samples. The second major grouping separated the European samples from the East Asian samples.
Oh, wow. That is the smoking gun.
It is absolute validation. This clustering perfectly mirrors known historical human genetic variation and migration patterns.
Right, because an algorithm's random error rate isn't going to accidentally perfectly mimic human evolutionary migration.
Exactly.
Yeah.
Think about the logic. If these newly discovered unmapped RNA splicing events were just random algorithmic hallucinations or sequencing noise, they would be distributed randomly across all 202 people. They would not perfectly reconstruct the map of global human ancestry.
That proves these unmapped junctions are real, inherited biological realities passed down through generations. They are physical traits dictating how our proteins are built, and they've just been sitting in the dark waiting for a tool smart enough to see them.
It demonstrates that longcallR is not just an incremental software update to make data process a little faster. It is a completely new observational lens.
All right, final recap here. Applied to 202 humans, the tool found widespread allele-specific splicing. Nearly 50% of these junctions have never been mapped before. And these unmapped junctions track perfectly with known global human ancestry, proving they are biologically real.
That's the whole picture.
It's like a flashlight that is suddenly revealing entirely new rooms in the architecture of the human genome. And reading the conclusion of the paper, it's clear the authors aren't stopping here.
No, their future plans are incredibly ambitious. They explicitly lay out their intention to adapt this algorithm for single-cell RNA sequencing.
Which is a huge leap, because right now, this benchmarking and population data was all done on what we call bulk RNA-seq. Bulk sequencing is essentially taking a chunk of tissue, putting it in a blender, and measuring the average genetic output of millions of cells all at once.
Exactly. It gives you the average state of the tissue. But moving this tool to single-cell sequencing means they will be able to explore cellular heterogeneity. They will be able to see how these intricate splicing events differ from one individual cell to its immediate neighbor.
Wow. So as we wrap up this exploration, think about what this means for you and for the future of medicine. If nearly half of these allele-specific splicing events in bulk tissue were previously invisible to us, what on earth are we going to find when we point this hyper-accurate flashlight at the level of individual single cells?
It's going to be unprecedented.
Imagine being able to track how a single unmapped splicing error in one specific cell line contributes to a complex cascading disease like cancer. We used to think we had the map of human biology pretty well drawn out, but it turns out we've just finally found the right lens to actually read it. Keep exploring.